Hello, everybody. My name is Chris Troll. I'm the Chief Scientific Officer for ClaretBio's, and today I'm going to be talking to you about our enzymatic DNA shearing module, which we call ForShear. But as a part of this presentation, I'm also going to be talking about FFPE in general, FFPE DNA in general, how to get the best data for your NGS, next generation sequencing from FFPE DNA. And while this presentation will focus on ClaretBio's and ForShear, I am really hoping that it'll be applicable to the general audience and everybody will gain some knowledge in general, even if you're not into ClaretBio's products. So with that, I'm going to begin with my convolutedly titled presentation called Shearing Your FFPE DNA for NGS For Sure with ClaretBio's ForShear. So I like to start my presentations by taking a huge wide angle and then narrowing down. So the widest angle that I can think of to start a presentation about FFPE DNA is talking about what FFPE is. So FFPE samples are important because they're a dominant form of preservation that pathologists use when taking a biopsy from the patient. And like all preservation samples and tissue samples, it's going to start with a biopsy, which is done with a biopsy needle or a scalpel, depending on the location within the individual that is having the tissue sample taken. After the tissue sample is taken and any sort of fresh analyses are performed, the pathologist will usually place the sample gently into buffered formalin, which is your stock standard liquid formaldehyde solution that is buffered. It is pH balanced, so that helps preserve tissues. It is, anywhere between 10% to 40% formaldehyde, depending on where you're getting your formalin. But dunking these tissue samples in buffered formalin is akin to all the museum specimens that you see when you go to the museum, all those old jars with like squids or octopuses in them. That is buffered formalin that is helping preserve these tissues for hundreds of years. So after the tissue has been fixed, and by fixed, I mean the buffered formalin causes cross-links and a bunch of other preservative actions for the tissue, then the tissue is embedded in paraffin. The paraffin itself helps by giving structure to the tissue. It gives rigidity. It allows also for protectiveness of the tissue so that the tissue block can be handled without the tissue damaged. It dries the tissue, which helps with cell staining and a bunch of other microscopic based assays that pathologists use. So this is the process for creating FFPE samples, and it has been around for a very long time. And it makes a lot of sense for a lot of the assays that pathologists use, especially assays that were done way in the past. And by way in the past, I mean what? 20 years ago. Not to say these assays are not completely valid today. They are. And all the microscopic assays and the cell stainings are done ubiquitously across time. But now with the advent of molecular technologies, there has been an additional layer of analyses that can be done, which is from your extracted FFPE DNA. That allows for, if this is the sample type that DNA researchers can get, that's great. It also, really importantly, allows for retrospective studies. These FFPE blocks can be stored for upwards of 40, 50 years. In fact, even people come to ClaretBio's because not only for FFP, but we work a lot with the ancient DNA communities that work with museum specimens and all sorts of fun stuff. By stuff, I mean fun animal and plant tissues that have been fixed and just sitting in buffered formalin for hundreds of years. So particularly if you're a DNA researcher, this whole process makes the DNA degraded. When you take the biopsy, you fix it in the formalin, you embed it in the paraffin, and then it's sitting there on the counter at room temperature, completely fine. when a DNA researcher goes to extract the DNA, this is what it looks like. And what you see here is a tape station readout. A tape station is just a type of digital fragment analyzer. These are your bioanalyzers, your tape stations, your fragment analyzers. All these will kind of give a similar readout. And so what you see on the left-hand side here is the DNA ladder, which just comes standard. You run it as a control alongside so you can see the size of your DNA that you're extracted. This is the size in base pair with 10,000 bases, or 10 kb being the upper marker and 15 bases being the lower marker in purple and green respectively. And you see this extracted DNA is pretty short, relatively speaking. And by relatively speaking, I mean compared to a fresh tissue or cell culture extract where the DNA is going to be high molecular weight. And so here you can see a difference between about the fresh DNA extraction, the fresh cell culture goes to 15,000 base pairs or 15 kb, probably even higher than this. And there's no real shorter molecules here, as you can see here. It's kind of all just one big mass of high molecular weight DNA, as we call it here. And here you get a smear. You can see there is some DNA that's higher. It's a gradient, right? So there's some DNA that's around 1.5 kb, around 1 kb, but the main mass is really low. And that's fragmented, and that's one sign of degradation. In fact, that's the easiest sign of degradation we can measure, but embedded in this fragment size based metric, there's also a bunch of other problems, so to speak, lurking for DNA researchers. And these problems are, DNA damaging lesions, single-stranded breaks, cross-links. All of these can cause DNA shearing, but they can also be left over in the DNA product post-shearing. So what you can see here is potentially single-stranded DNA breaks, DNA damaging lesions causing the DNA to fragment upon purification out of the fixed sample. But what you don't see in this gel trace, this tape station trace, is the remnants of cross-links and DNA damaging lesions. Hopefully you've reversed some of the cross-links, the majority of the cross-links actually, when you extract the DNA, because there is usually a high temperature setting for a certain amount of time that can be really painfully long, 4 hours or overnight. So at a high temperature, 55, 65 degrees, that's supposed to reverse all these crosslinks, but there can be remnant damage from those, and not all the crosslinks can be reversed, because as we all know, no real biological assay or event, like a ligation event, a polymerization event, really goes to kind of 100% efficiency. So I think that's true in extraction. So while you can get a DNA integrity score from this, I think there are also other factors that contribute to the quality of the FFP extract that you're getting. Whether it's high quality or low quality can't be judged solely on the basis of your kind of extraction method here. All this is to say that these problems lead to difficulties. Most of the difficulties are going to be in PCR. That is because if the polymerase encounters a extension polymerizing blocking lesion, then the polymerase just kind of gives up. It's like, I can't go past this modification. So then that molecule drops out from your NGS next generation sequencing library, and that results in poor conversion efficiency and less complexity for your library. This can then in turn lead to poor data where you don't have enough reads covering the specific loci that you want in order to call your variants, your SNPs, your word I'm blanking on, it's not SNPs, but your chromosomal rearrangements, fusions. Thank you. That's the word I'm looking for. It makes calling these potentially deleterious events or potentially driver mutations, if this is a cancer sample, difficult to call if you don't have enough data, and that can just lose your sample or have inconclusive data. Another fun aspect of FFPE DNA is its complete variability. And this comes from a variety of conditions, steps in the process, but there is no standard method for how to create an FFPE DNA, an FFPE sample for pathologists. The fixing in the buffered formalin time can be variable. The amount of drying time and paraffin embedded can be variable as well. And also, not to mention, these samples can sit on the shelves. One sample could be 6 months old, another sample could be 20 years old. And that's going to have an effect on your DNA quality. So you can see here, these are literally just 4 different extracts from 4 different prostate cancer FFPE samples from a prostate biopsy. And you can see the variability in size here. So this is a genomic DNA tape, which is why it goes all the way up to pretty much 50 kb here, and at the low end, 100 base pairs. So the variability in size here is quite a bit between these samples, of between 1.5 kb here to 4,000 kb here. And you can tell that this one and this one particularly even has DNA that's pretty high molecular weight. It has some molecules, all of them have some molecules that are 50 kb and some molecules that are lower size. And but you can obviously see where the mass is there. So how to get the best data from your FFPE samples? More particularly, how to get the best next generation sequencing data, the best next generation sequencing libraries from an FFPE sample. So we're talking, at this point, you have identified the FFPE samples that you want. You've gotten them. You've extracted the DNA. You have QC'd the DNA. Always QC the DNA with both a tape station or any sort of fragment analyzer, as well as for the concentration that you have extracted, whether that be nanodrop or qubit. Most people these days use a qubit or some sort of fluorometric reading. It is more accurate and less susceptible to leftover residue within the extraction, such as ethanol or whatnot. So the one thing you can't get from a qubit is your absorbance rates, but a lot of that stuff doesn't matter as much for NGS library as it did in the past. But extract your DNA, get your metrics, your size, and your concentration, and then your next step is to shear your samples. And there are 2 reasons to shear your samples. One I've already alluded to earlier, and I'm going to come around back to that point in a minute. But first, the simplest reason to shear your samples is to get a uniform size, a size ideal for sequencing, and this is based on the Illumina sequencers, which are pretty much akin or synonymous with the words NGS. The synonymous with the initialism NGS. So what you can see here is some of those prostate FFP extracts that I mentioned in the previous slide that are variable size. And then you can shear these either enzymatically or acoustically. These were actually acoustically sheared, and now they have a uniform size with a mass centered around a good size for bridge amplification, which I'll talk about in a second. But another thing to note here is that the size of this gel is a genomic gel, which goes up to 50 kb. And this is a D1000 gel, which, despite its name, goes up to 1.5 kb instead of just 1,000 bases. But things get squished at the top of the tape station specifically, so it's justified that it's called a D1000 is all I'm saying. And so the size here is about 250 bases, which is pretty ideal for shearing an FFPE sample. And this is way outside the range for NGS of an FFPE sample. So talking about the 2 reasons. The first reason is simply due to the constraints of the sequencer. This is not an issue, potentially, this portion of it, for 3rd generation sequencers that are your longer reads, which are your nanopore and your PacBio machines. But I don't think FFPE is a good sample type for those machines, and I'll explain to you in a second when I talk about the second reason. But back to the point, Illumina flow cells perform bridge amplification on your DNA inputs once the libraries load onto the flow cell. And this causes the clusters so that the sequencer can read your samples and read the libraries and the molecules from the sample via extension. Bridge amplification really likes short molecules. In fact, the propensity to bridge amplify is inversely correlated to the length of a molecule. That's not to say that molecules longer than 500 to 800 base pair won't bridge amplify. They could, but they're just going to be outcompeted by shorter molecules, which is why, for all intents and purposes, say about 500 to 800 base pairs is a good max fragment length, so it doesn't get completely outcompeted with, dimers or smaller libraries if you're running a mixed sample pool on your Illumina sequencer. So 500 to 800 base pairs max fragment length is good. And that's probably good for any sort of high quality DNA. But as I mentioned, FFPE has got a bunch of lesions in the DNA that are lurking there. So, and that is not seen in the view of this tape station. Sure, you could do assays such as fluorometric assays to look at your abasic sites that are in there, or oxidative lesions or methyl lesions, but who's got the time for this? Like, we just want to extract our DNA, we want to make our libraries, we want to enrich it in our panels, and we want to shove it off to the bioinformaticists, so it's their problems to call the SNPs, and we don't want to deal with it anymore. So for that reason, the second reason to shear your FFPE DNA extracts is the shorter the fragment, the less likely you are to hit a polymerase blocking lesion within that set of fragments. So by that, I mean, let me explain this in the reverse way. The longer a fragment is, the more likely you will hit a polymerase blocking lesion in that fragment compared to a shorter fragment. I'm going to let that sink in. Okay, I'm done. So basically, the shorter the fragment is, the more fragments that it's possible for the polymerase to completely amplify through, which is great because in NGS library preps, you need your P5 on one side, your adapter. Some people call it an I5 index, index sequencing primer, whatever you want to call it. And on the other side, you need your P7 index sequencing primer, I7, P7, same thing. So you need to go all the way through the template on your polymerase PCR cycling extension. And if you don't get that, if there's a truncation in the middle, you'll lose your P7 or P5, depending on which way the thermocycler is cycling your molecule, and that molecule will drop out. Less likely, obviously, as the non-input molecules build, but during the first cycles of PCR, this is very important. So therefore, the second reason I believe it's important to shear your FFPE samples is just because you are competing with the odds and probably bettering your odds in order to have more complexity of your libraries there. So, end of that rant. Let's move on to what about lower molecular weight FFPE samples or poorer quality of samples. So if these samples, I'm going to go back a slide right now to show you, but if these samples are considered higher quality or good quality, as I've listed here, because simply by the tape station just saying that these molecules are of a higher molecular weight, these are poorer quality samples because my tape station tells me that the mean the mean extract size is about 250 to 400 base pairs. So should you shear these samples? Well, based on everything I said, for at least the second part of what I said of the polymerase blocking lesions, yes, you should. But what about the first part for bridge amplification? Well, you can definitely make libraries out of these without shearing your samples, but our data shows that shearing even poor quality samples really helps the complexity of the library. So what you're seeing here on the right side of your screen is a pre-seq library complexity estimate. So what this output shows is the methods for this are these samples have been taken and put through a library prep, and they have either been sheared or not sheared during the library prep process. Then they have been sequenced to a certain number of reads. And then this computer software extrapolates the number of reads it's seen, the number of duplicates it's seen, to give you an estimate of how many unique reads are in your library for this particular sample. And it's the caveat here is it does this on a very low amount of reads. So basically you can sequence your samples to, 2 million reads, 1 million reads, just to make sure that your library is of good quality, that you've got good mapping rate, that you've got low duplications, you've got low chimeras, nothing, there's no contamination in there. It's all mapping to human or whatever organism you're extracting your FFPE samples from. So you can do your quality control check so that you can convince yourself that this sample is good enough to spend more money on, basically, because the more number of reads you sequence, the more expensive it gets. So you want to make sure your sample looks good before you spend the thousands of dollars to get it on a NovaSeq, or even maybe you want to do this before you enrich your sample to make sure it shows that you have enough good quality reads, good quality molecules, so that enrichment via your exome panel or your targeted cancer panel is worth using for this sample. So that's all this is. So what you see here is basically the way this works is if you were to sequence 100 million read pairs, 200 million read pairs, or 300 million read pairs, you would go up this line, and this is how many unique molecules you would expect to see, given how many you sequenced. And you want the ideal line is an X equals Y line, which just is, a straight 45-degree line, but that's, completely theoretical and never happens because you don't get infinite complexity from samples. So there's always some amount of duplication in there, unless maybe you're doing PCR free, but even that's got some caveats to it. But we're not talking PCR free. We're talking about ligation based amplification, normal stock standard NGS algorithms. So here the sheared ones are always more complex. And this is due in part to both reasons. One, if you shear these to a uniform size, all of these molecules that are up here that are kind of outside the optimal bridge amplification range all get compacted down here into your average size. And then furthermore, your average size of your main mass of your extract will move down a little. So before I move on, I just want to say that it is a delicate balance. Obviously, you don't want to overshear your samples. You don't want them too small. You still want to be able to map. You want to be able to use all the read length in your sequencers. So shearing somewhere in that 150 to 300 base pair fragment range is usually your best bet. Anything smaller than that, and you kind of get a hard time mapping. You also get a hard time enriching, since enrichment probe panels really prefer longer molecules. And by longer molecules, I mean greater than 100 base pairs, 150, 200 base pairs or so. And also, on the converse of that, you don't want it too big. So yes, shear your samples. It's going to do you a world of good. Doesn't matter if they look like they don't need to be sheared, they still need to be sheared. If they look like they need to be sheared, great. Why do we have this discussion in the first place? Just kidding. Let's move on and change subjects a little and talk about the different shearing different ways there are to shear FFPE DNA, or actually different ways there are to shear any DNA that's out there. And there's two main ways that the community has settled on, and that is your acoustic methods, these are your machines, and your enzymatic methods. So acoustic, the main forerunners in this are as pictured here, a Kovara shear, or shoot, I forgot the name, but it'll come back to me. Oh, diagenode bioreactor. Sorry, diagenode. So Kovaris or diagenode, those are your kind of two main machines in the space of DNA shearing right now. And enzymatic, every library prep company and their mother, worth their weight in gold, has a enzymatic solution. And you'll see why here in a second. In fact, it's already here. So let's just briefly go over the pros and cons, but spoiler, the main pro for enzymatic is just that it's cheap and everybody cares about it, which makes sense. So the pros, so let's back up. The pros to acoustic shearing are that there is minimal sample loss because what's in the tube stays in the tube. It's hard to overshear due to the biophysics of a DNA molecule kind of flexing like so. They will only fragment if they're a certain length. Think of like a spaghetti noodle. If it's a long spaghetti noodle, it'll break. If it's a short spaghetti noodle, it's going to just kind of flex, but it's not going to break because it's too short and it doesn't have that like wavelength to really just like crack it apart. So that's what I mean by hard to overshear. You can blast samples in a acoustic sonicator for a long time and you'll still not lose a lot of your sample. Of course, the cons are that it is a machine. And as we all know, machines in the biotech world are just expensive. Doesn't matter what the machine is, even if it's just a normal thermocycler, which has been around for 40 years, they're expensive. So a dedicated machine that does a specialized process is going to be expensive, and a DNA shearing device is no different than any other specialized machine in biotech. Another con is that it can be time consuming to batch samples. So if you have 10 samples to shear, depending on the machine you buy, some machines only shear one sample at a time. So what you're left doing, put the tube in, push the button, shear, wait, flip through your social media, take it out, do the next one. And the number of samples just, logarithmically kind of accumulates in terms of the time you're sitting there. Some machines can do more in terms of the number of samples they can batch and shear, but C point one under cons if you think a single shearing machine is expensive. So moving on to the enzymatic methods, the pros are that it's cheap, it's easy to batch samples because it's in a 96-well plate. It's just a liquid in it too, is all it is. So it goes with normal pipetting. You can pipette it with multi-channel, you can automate it, you can, batch in 96-well plates, 8-well strips, doesn't matter. The cons of the enzymatic method are basically two. There's a third one here, which I'll explain in a second, but sample loss. Enzymatic shearing enzymes are hungry little devils. They like to cut DNA. And so it's a delicate balance between under-shearing, over-shearing, and if you let it go too long, the worry is that an enzymatic shearing method can chew up all your DNA that's in there. That is not necessarily the case because different companies have come up with different really intelligent ways to kind of mitigate that. So it is less of an issue now than it was when enzymatic shearing methods came around, but it's still a viable concern. And if you can overshear, you can lose your sample. They're kind of one and the same. The third con here is that there's enzymatic bias when you shear. Acoustic sonicators don't have much bias. By bias, I mean they'll see any DNA sequence and they'll just kind of cut because it's more of a physics thing rather than a sequence dependent thing. So because of that, there is no real bias of whether it cuts at an A, C, G, or T at its cut site. Enzymes have preferences. They're biological machines. And as biological machines, they have recognition sites. And so therefore, they prefer to cut at certain bases or not. Whether this issue is valid or not is completely up for debate, but you definitely don't want something that's overly biased. But you have to accept some levels of bias when you're talking about this. All that being said, most people prefer enzymatic methods for the three reasons listed under the pros. Cheap, easy to batch samples, easy to automate. Usually that wins out, especially for labs that aren't dedicated genomics labs too. If you just have some samples and you're trying to do, an auxiliary or ancillary study to what you normally do. You can buy a kit with an enzymatic shearing method. You can make your libraries, you can send them. It's just a lot harder to access those if you don't have access to them already. By them, I mean acoustic or mechanical signatures. Okay, that is the big intro to the presentation and very ClaretBio agnostic or seriously agnostic. Now I'm going to get into the part of the presentation that's a little bit more marketing or commercial, as you could call it, but I hope that there is still some great information that you as a researcher and as a potential customer can gather from this part. Basically what I'm saying is don't tune out now. We've already gone through like half the presentation. So just sit through it. There's still some interesting stuff. So that being said, let's talk about Seriously. Seriously is ClaretBio's answer to NGS library prep kits. So it is, you know, our kind of special version of a method that gets you DNA onto an Illumina sequencer. A solution that most companies that are in the NGS space, the biotech, the library prep space, your NEB, your Takara, your Twist, your IDT, all of these people have solutions to this. This is just our solution to it, and it's called Seriously. It's a single-stranded, single reaction library preparation method. It is, yes, we did shoehorn that acronym into what it stands for, but that's okay. We're okay. So our method is a little bit different in the way the adapters are delivered to the template DNA. So all template DNA pools start out as kind of a complex mixture of DNA. And the mixture of the DNA depends on the input DNA, the template DNA, the extracted DNA. For example, if you're getting your DNA from a high quality source, such as that cell culture, that fresh frozen DNA, you are going to have more contiguous fragments with either overhangs or blunt ends, and less single-stranded or nicked or damaged stuff being DNA molecules. Whereas if you're extracting from an FFPE sample, you may have more damaged double-stranded DNA and single-stranded molecules than you do contiguous, beautiful double-stranded DNA. But it just depends on the sample type. If you're working with, you know, single-stranded DNA, your sample type is only single-stranded DNA. This would be, for example, your cDNA from RNA-seq, or if you're doing oligo QC, or any sort of probe-based QC of your enrichment panel. So anyways, this pool varies, is all I'm saying. So once you have your pool, which isn't really part of the Seriously reaction, you have your pool, you put into Seriously. The first step of Seriously is that all the molecules are denatured to be single-stranded. So contiguous double-stranded molecules become two single-stranded molecules. One single-stranded molecule just stays as one single-stranded molecule, and a double-stranded molecule with a nick in it or a single-stranded break. I'm using both terms synonymously. Those will become three molecules, one for the bottom and two for the top, with the nick being the shorter of the two strands. So the first step, denature all the DNA, add our single-stranded enhancer proteins, which kind of bind to this single-stranded DNA and keep the DNA single-stranded, prevent it from going back to itself, annealing back to its complementary partner, preventing it from creating secondary or tertiary structures. And then after that, you ligate our adapters. So the adapters here are shown in yellow and green, I believe, teal, maybe a yellowish orange. So the deal with these adapters is these are duplex adapters. This forward adapter is your P5 adapter or your I5 adapter, your forward sequencing primer, whatever you want to call it. The reverse is the I7, the P7, your reverse template sequencing primer. And so those are your forward and reverse. The forward is a double-stranded duplex where it is Watson-Crick base paired from the top strand to the bottom strand, which is not documented here, but except in its close proximity to the top and the bottom. This is a inert splint, as we like to call it. We call these splinted adapters. splinted adapters have been around for quite a while. Splints are really cool and amazing pieces of single-stranded DNA that have multiple purposes and functions, particularly in biotechnology. They're not probably found so much in nature, but don't quote me. But the reason this is a splint is because right here, where the splint molecule does not overlap with the forward primer, but overlaps with the template itself, which is in blue. These are just random bases. So these random bases basically create a localized double-stranded annealing product such that T4 ligase can ligate the single-stranded DNA to the double-stranded splint adapter. So these splints just facilitate your standard double-stranded ligation, but in a cool, unique way because it's localized. And then afterwards, these splints kind of go away because they don't take part in index PCR. These red stop signs are meant to say that there are just ligation and extension blocking lesions on all termini of the splint adapters that you don't want to participate in the ligation event or into the subsequent amplification. This is what makes the splints completely inert, because they cannot ligate or amplify, and the directionality of the adapters is such that they will only ligate in the proper orientation to the template itself. So after ligation, it's just simple PCR. This PCR is stock standard. As we all know, in PCR, the first step is initial denaturation. So whether you're performing seriously or standard double-stranded prep that's got the end repair, the A-tailing, the adapter ligation, then PCR. Once all those library preps end up at the initial denaturation of index PCR, they're all exactly the same because you have individual single strands and your amplification starts with the P7, the I7 index primer coming in and just amplifying across this way. The first cycle of PCR is exactly the same for seriously as it is for any other Illumina NGS library. And what this means is at the end of seriously, you get a standard double-stranded library that is compatible with Illumina sequencers, that is indistinguishable from any library prep by any other company. So it is completely compatible with any sort of third-party downstream enrichment kits. It's compatible with any Illumina sequencing kits, any Illumina sequencer, etc. The only thing that differs between seriously and a more standard double-stranded prep is just the way in which the adapters are compelled, attached, ligated to the template. So we've got a lot of other, nope, never mind, talk about that in a second. So let's talk about the enzymatic shearing module. So ForShear, which is kind of the point of this whole presentation. But basically, the only difference between a regular seriously, vanilla seriously, and ForShear seriously is that the input differs between the two. For regular seriously, you are assuming, and I'm going to go back 2 slides right now, that this DNA is already sheared or has come in a format in which it does not need to be sheared. Certain sample types that do not need to be sheared, which are not discussed above, because all FFPE samples need to be sheared, right, are cell-free DNA, short oligos, ancient DNA, those kind of samples, forensic DNA, anything that is very, very naturally short. MicroRNAs, those don't need to be sheared. Those are super tiny, right? 21, 22 nucleotides, super tiny. Okay. So this assumes your input is genomic DNA, FFPE DNA, any sort of extracted tissue-based DNA. So the ForShear enzyme then shears the DNA to the right size based on the parameters within the methods, and then it's your stock standard seriously. So after you ligate, you then amplify. This feeds right into this, which feeds right into this. Fragmentation feeds right into ligation, which feeds right into amplification. So now I think I can talk about the other thing. Here it is. Yes. So we have an optional UMI extension step. This is not necessary for creating your libraries. You can do without this kit. But if you want the ability to call low allele frequency variants, then you're going to want this UMI added step. it is an extra step in the protocol. It's an extra scry clean. It adds probably 45 minutes to the protocol. And what you get is a UMI embedded in the I7 index, which can then be sequenced via your I7 index read and deconvoluted via some software we have on our website or some free open source software you can use. The UMI gets eventually placed into a tag in the BAM file, and then you can use this when you're calling variants or whatever else you're looking for, resolving PCR duplicates. If you have a low complexity sample, such as you're looking at oligos, then you can use UMIs to resolve what's a unique molecule from what's a duplicate, or what's a variant from what's a sequencing error. So let's go further into ForShear. So ForShear has a few benefits to it. And the first benefit is that it's fully integrated into Seriously, no additional purification step. This I already discussed. Let's go through it one more time, just for the sake of the fact that it's in the presentation here, and it'd be awkward if I didn't go through it again. So high molecular weight DNA is then sheared. The shearing goes straight into Seriously adapter ligation. Only after ligation is there a purification step via DNA purification beads. We sell them. Everybody else sells them. Doesn't matter whose you use. They all work well. After you purify, the purification via DNA selection beads also helps you size select out your desired product from nasty things like adapter dimers and short DNAs, which just aren't going to map well anyways. Then you can amplify via index PCR enrichment. So it is fully integrated into Seriously, which is great. Less cleans are always better. It shears both double-stranded DNA and single-stranded DNA, which is super neat. It is a little less of a worry because single-stranded DNA doesn't normally come in a very long product, except for very special circumstances. There is long, obviously RNA is long, but that's kind of sheared in its own process when it's converted into cDNA. But to exemplify the effect that ForShear shears both double-stranded and single-stranded DNA, here we just have a single-stranded or double-stranded oligo that's either one part of it or it's annealed together with its complement, and then it's put through ForShear Seriously, and it's lightly sheared. So you can see that the product itself still remains, but you also have sheared products below that. If you ever want to do this with oligos or any other, if you have longer oligos, maybe, or if you're looking at some longer RNAs for, sorry, longer DNAs for, you know what, I'm not even going to try to say what's out there, but biotech is a big field, and I'm sure there is an application for this. So it shears both double-stranded and single-stranded DNA. It's not necessarily, you know, something that we advertise because it's not used that much, and it's not something we were trying to develop when we developed it. It just happened to be that it shears single-stranded DNA, which is a nice benefit, so that all your molecules contain the same size. Moving on. ForShear is robust, very difficult to overshear your samples. So for example, let's take a FFPE DNA extract that has an average size of about 600 base pairs here. You can see that the mode is much shorter, but because there are higher kind of molecules in here, that draws the average size up. And this is a D5000 tape station, for those wondering, with the ladder on the left, and you have your 100 base pair fragments all the way up to 5 kb. So it has robustness in terms of shearing time. In the protocol, we tell people to shear for 5 minutes. However, if you forget your sample, or if you're going to get a cup of coffee and this is in the morning, or you go to eat lunch and you forget for a few minutes, it's not that big of a deal. Your sample will still be fine up till at least the 30-minute, which is, you know, that's pretty good. It's pretty forgiving. You know, that's 5, 10, 15, 10, 25. It's like 6 times longer than it's set in the menu. The other thing is there is uniform shearing in the DNA inputs amounts. So the range of our seriously kit is about 5 nanograms to 50 nanograms. We can get good variant calling within that range from any FFPE sample. Generally 50 nanograms is more than enough. Sometimes FFP extracts are low, but usually not low enough that it drops below kind of the 10 nanogram range, but they all shear uniform. So with 5 minutes of shearing, you can get 50 nanograms of DNA down to 10, the same size as 10 nanograms of DNA without any loss in. So let's talk a little bit about enzymatic bias now for ForShear, because I think enzymatic bias, or at least looking at the end profile of molecules, is fascinating, mainly from our research into cell-free DNA, which I'm not talking about here, but it's just kind of a nostalgic feeling, and I find it very interesting where the biases and shearings are for different molecules. So basically what I am talking about is the point where the enzyme cuts. So let me orient you here. So what you're looking at is 2 plots, and they are not mirror images of each other. They are different. This is the read 1, and this is read 2. And then, so at this 0 point in the position, this is where the read ends, and the genome that it maps to, sorry, not the genome, but the upstream region in the genome that it maps to starts. So the actual read maps to the genome, if we're going 3' to 5', which we probably shouldn't because that's not the way biology works, but we're going to do that anyways. If we're going this way, this is all read, this is all read, this is all read, this is all read. The read stops, but we know the context because this read maps with high quality to the human genome, the human's reference genome. So we know what base starts up the actual genome that's kind of upstream. And so that's what's meant by genome. And the same thing here. So read 2, this is going 5' to 3', blah, 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 stop. For the read itself, and then the genome that's here. And what you have here is basically the base tendency that is seen at this cut site. So these are log2 odds, which means that anything that is at 1 is roughly twice as likely to be seen than something that is 0. So you see here that ForShear has got some bias. As I mentioned, all enzymatic shearing modules have bias within them because they are biological machines. So yes, ForShear tends to cut A's, cut at a location where there is an A, more so than a location where there's a G, more so where there's a location than a C, and it likes to cut least amount at T's. But it cuts at one site because the way it works is it is an endonuclease, and it just makes a straight kind of blunt cut, which you can see here because it just cuts across this way. This is, and the reason I mention this is because this is opposed to other shearing enzyme cocktails. So other enzymatic shearing cocktails usually use a nickase that cuts the top strand, and then another nickase, I guess, but this other nickase recognizes where that first nick is and then finds the spot that's kind of around there and cuts again. So it's kind of a dual process of like one snip, second snip. And that's what you can see here. You can see that there is kind of variability in the snipping, in the cutting. So maybe the first cut is here, and maybe the second cut is here, or the first cut is here, and the second cut is here. It's dynamic. This is kind of integrated over the whole reads, the whole genome. So it's hard to suss out any kind of, there's no real, what's the word I'm looking for? There's no motif that we're looking for. There's no specific motif. If there was an actual motif, it would be a bad shearing enzyme. But there's a degenerate motif or a signature, so to speak. And so the difference here is that ForShear is more of a clean cut across, and this is more of a staggered cut. And what this leads to is that, as I already talked about, the ForShear cutting A. So here, oh, maybe it needs to cut it cuts first at a C, as you can see here, blue, and then at a T here. So this kind of complex cutting, which you can see here, is that there is some sort of variability within the what kind of follows in the read. Oh, the other thing I should mention is that this is just looking at read one for simplicity's sake. And this is what was actually getting my head a little messed up just right now when I was talking about it, because I was thinking about the last slide of this being read one and read two. I'm like, why is this in here? No, this is genomic. This is the read. So this variability, this more stringency in the cutting here leads to a little bit more variability within where the read is. This settles out over time. Don't get me wrong. Once you go past this into like the 15 bases into the sequence, it kind of goes more uniform here. But there's a little bit more kind of end bias here in some other enzymatic shearing enzymes than there is in ClaretBio for sure. As you can see, this kind of goes back to the baseline, which is more similar to the genomic region than this is. There's kind of a less similarity here. There's just more stringencies involved in the shearing enzyme. Now, do I think this really matters? Not really. I think it is a nice benefit, and maybe it matters if you're doing some certain applications or don't have a lot of complexity. But overall, a long read length, a lot of input is going to overcome any of these enzymatic biases. And I don't mean that just for ForShear compared to other enzymatic shearing enzymes. I mean this in terms of any enzymatic shearing enzyme worth its kind of weight in salt. Not its weight in salt, but any commercial enzymatic shearing enzyme that's out today, it's not going to matter compared to an acoustic sonicator. Yes, acoustic sonicators do have less bias, but it's easily overcome by the amount of input, the complexity, and just the long read length, where you're gonna see these. So what if it doesn't cut at this one base and it can only cut like 5 bases downstream, right? Or upstream, you're gonna get all of that information in your massively parallel sequencing. Okay, I have talked way too much about enzymatic bias. Let's just move on from there. But seriously, it has an interesting enzymatic shearing profile, and it doesn't have a lot of bias upstream and downstream. Easily automatable. A lot of enzymatic shearing modules are easily automatable. Seriously, ForShear is no different. In fact, even to help you with your enzymatic shearing high throughput automation needs, we have software and scripts available for download for the Agilent Bravo, for the Hamilton NGS Star, for Operons. We are working with other companies as well, and we're always happy to have feedback, and anybody who needs help with scripting for their automation. We like to build our library of automation scripts. I think it helps us as a team and a company, and helps our customers as well. So reach out to us if you have any automations. We'll do our best to help. So what I've talked about here is embedded within our Seriously Kit product page. This is what you're seeing. This is what pops up on the website when you go to order the kits. And what I've been talking about is basically the Seriously NanoPlus kit mixed with the UMIs, UDIs. You can also just go with the normal UDIs. UDI stands for unique dual index. They don't have UMIs. UMI stands for unique molecular index, unique molecular identifier, mixed with the unique dual index, the dual index you need for the sequencer, with our DNA purification beads, and this is the ForShear. So just going real quick, in order to show the validity of our ForShear module and Seriously and its power for calling variants, we had a very simple study that was set up where we used the Horizon standards. These are FC DNAs, that stands for formalin compromised DNA controls, and they come in 3 different flavors: mild, moderate, and extra spicy. Just kidding. By extra spicy, I mean severe. So severe formalin damage, moderately formalin damage, mild formalin damage, to mimic the different states in which you receive your normal FFPE samples in. They've got a host of mutations in there, but 11 of them are quantified by digital droplet PCR, and they're at variant allele frequencies. And you can see the size of these if you just take the standard that is given to you from Horizon and quantify it via tape station. This is a D5000 tape station, so the mildly damaged formalin control DNA is formalin compromised DNA is high molecular weight, and then about 2 kp, and then really about 250 base pairs or so item size. So if you let's just take the two extremes. Let's not worry about the middle ground. The porridge is either too hot or too cold. They're not talking about the porridge being just right. That's a horrible, horrible analogy. I don't even know why I said it. Talking about just the severely damaged FC DNA control and the mild one. I'm just saying we excluded the moderate. Sorry. Then you put it through seriously, little asterisk here, which means we used our UMIs, enrich them with an exome panel, sequence them on a 2x150 base pair run, and then run them through our software. Software available on our websites are Seriously UMI and Seriously Run. Seriously UMI helps with the UMI demultiplexing based part of it, and Seriously Run helps map the reads to, I believe it does BWA and NEM. So, and outputs some other metrics for you. So, just some molecular results for you here. You can see that the shearing worked quite well on the mild sample. If you remember from the last slide, it was about 10 kb or bigger, and now it is at 250 bases. The severe was at an average size of about 250 to 300, and now it's a little bit smaller, but see my slides above about how Seriously is robust with respect to shearing of input and size. And yeah, you can see that, as expected, the severe DNA is a bit smaller because it started off smaller. It probably has more lesions. So there's more of a selection bias towards shorter molecules getting through PCR. And you've got enough yield with minimal cycles in order to do downstream, which is what we did. And this is the level in which we sequence these. So here is the mapped insert length. So this is post-sequencing by taking the kind of mapped reads in your BAM file and just looking at the distance between the start of read 1 and the start of read 2. So the start of the molecule and the end of the molecule. What you get is you see that the severe one is shorter. The sequencing is an average size of 100 bases. The mild is longer. It still has a mode around that 120 bases, but it's got a much longer tail that goes out to 500 bases. This is just the percent reads at length that's integrated over this whole area. And the amount of reads that we sequenced. So, the depth that these were sequenced was, in terms of human genome coverage, was 14x and 11x. This is more for the whole genome sequencing prior to enrichment. Post-enrichment, these were sequenced much deeper. This is kind of just to show you the general metrics for this. Not the general metrics for the variant calling, which we'll show in the next slide, but just the general metrics. The point here that I want to make, though, is this final column. This is the read pairs required for 1x coverage. And you can see here that it requires more reads, about 10 million more reads for the severe to get to 1x coverage than for the mild. And so why is this? It has to do with both the read length of the sequencer that we chose, as well as the insert size of the DNA itself. So as you can see here, the severe DNA has a average size, a mean size of 100 bases. We sequenced this 2x150. So 2x150, if you go 150 bases this way, 150 bases this way, that's 300 bases. Most of the molecules in this sample are not 300 bases long. At the very most, it looks like they're about 200, 250, if you want to be out here. So that means if you're sequencing this molecule here, way over here at the far extreme of how big these are, you still got 50 wasted bases because you sequence 300 bases, but the template molecule is only 250 bases. And that's in the best case scenario. Mostly the template basis was the template DNA was only 100 bases long, and you sequenced 200 extra bases, which were meaningless. Not so meaningless when you go to the mild because you've got, more fragments that you can see the whole kind of length of the DNA. All this is to say, be mindful of the length of the input DNA that you put in. This gets a little bit harder when you batch samples of varying length in terms of the final library size or the input template size, depending on how you want to talk about it. But for this, if you were sequencing just the fcDNA severe control, it may be worth only doing a 2x76 run rather than 2x150. 2x76 is going to give you 150 bases in each way, which is going to see the full molecule, excuse me, of the vast majority of molecules that are in there, but not for the mild. Whereas if you're grouping higher genomic ones and you want to see more of your data, then maybe it benefits you to sequence 2x150. That being said, Illumina sequencers are pretty restricted in their size length that they allow. We're talking a difference of 150 total bases really versus 300. There's 2x76, 2x101, 2x151. Why do I say that extra base? Because normally it takes an extra cycle to calculate the base call at the previous cycle. So although it's 2x76, it's really, you just get 75 bases, 75 bases. Of course, they call this 150 cycle kit. It's because you can allocate those reads however you want. You could do 1x150, or you could do 2x76. You could do you could be a complete psychopath and do like 1x120, and then the other by 30. Forward read 120, back read 30. But that's for you to decide. Okay, moving forward, library complexity from these runs looked really good. This was, again, the pre-enrichment. You only really want to do this pre-enrichment because the complexity is going to decrease a lot with enrichment, because that's kind of the point, because you're doing targeted sequencing. What's really nice here is that seriously does really well with damaged samples. It's kind of our thing at ClaretBio, is to work with degraded and damaged samples. So you can see that despite the damaged nature of the severe, there's hardly any drop in complexity between the mild and the severe here, at least out to 249 reads. It looks like it diverges a little, but it's not going to diverge that much, even if you kind of go out to your standard lane of a NextSeq 2000 or so, 400 million, which is supposedly 30x. And then the GC content is great. The normal human GC content range is about 27 to 42 or so. So this falls right in that range. Okay, so to wrap this up, finally, when you enrich these two samples using an exome panel, you sequence again, you get all the metrics, and then you look for the variants. You can calculate and you use your UMI kind of base demultiplexing, your UMI error correction and variant calling. You get a very nice kind of correlation here of the digital droplet PCR range versus the calculated real allele frequency via sequencing. So you can see here that all of the expected frequency is pretty much in line with each other. The sequencing missed one call here, which is the lowest variant, the lowest allele frequency. But that was only because of the software we used. When we went back and looked at it manually, we did notice that it was there. It just wasn't at a high enough frequency to be called by, I believe we used low freq was the name of the variant caller we were using. But for all intents and purposes, we missed this because in a real clinical sample, you miss what you don't see. And so then the ForShear severe one was very good as well. We missed a few more that are in there. I believe if we had higher sequencing depth for this one, because it suffered from the same issue post-enrichment as it did pre-enrichment, we could have called these as well. But still, very good correlation. So I have rambled on for very long, and I am just going to give a conclusion here that says shear your FFPE DNA extract prior to or during library prep. It'll aid you no matter what library prep kit you go with. We have a shearing module at ClaretBio. It works well. I've shown you that. If you would like to try it, please let us know. And seriously works well with FFPE samples. So with that, I'm going to make a very quick announcement that if you go to our website, which is shown here, and go to the featured literature, you can see more of the data on that final thing that I showed you. By the final thing, I mean the variant allele frequency. We have a white paper on it that shows more of the metrics and more of the enrichment metrics that I didn't get into today. So please check that out. And finally, I want to mention, and I will talk about this in a future video, that we have a really cool RNA-seq module with ribodepletion that is out now. Look for a new webinar coming out in the future about this, but yeah, it is right here. New, really, are none. So it's not DNA-based, it's RNA-based. It does ribodepletion with probes. It does it in a very unique way. Being that cDNA synthesis happens first, then adapter ligation, then depletion. So you're not handling RNA for a long amount of time because RNA is prone to degradation. And we found that it works really well for ribodepletion. It's interesting, the probes are flipped from standard RNA probes for depletion during the RNA step. It's a little bit quicker, it's a little bit more robust because it's DNA, all that stuff. And it does well with general metrics, including kind of uniform gene coverage. No surprise, it works well with FFPE samples and RNA as well, which I'll talk about in a future presentation. But we also have a white paper on this as well. So if you go to the same spot, you can find it there. So with that, I'll just say thanks to everybody at ClaretBio, and this is who we are. You know, our motto is to give you the best data from your worst samples, because we build tools that focus on degraded DNA and degraded RNA. We're located in Santa Cruz, California, right below the San Francisco Bay Area. If you're ever in the area, it's really nice. Come by and say hi to us. Go to the Monterey Bay Aquarium, go to the beach, go see some sea otters. And yeah, it's a nice location. There are worse places to be stuck. We are ISO 9001 certified, soon to be 1345 certified. And here's a picture of us at our recent outing to the beach. We're a very small team. There are some team members who couldn't make it. I'm sure the Olympic logo will not be dated in any way when you see this presentation in a year or two from now. In fact, it'll come around again in 2 years for the Winter Olympics, and again for 4 years in the next Summer Olympics, which is in LA. And there were some people who couldn't make it, so we like to have fun. So with all that, thank you for joining.